So to start today's lab, as you saw in the lecture, we are introducing sequence alignment in more detail. Since in general, it's been mentioned throughout the course, just as the ideas of reads and sequencing have, but there's also a lot more to it, and it really is a fundamental part of bioinformatics. So to start, for this lab, we'll be doing some very, very simple read alignments to dip your toes in the water. So to start off, we have prepared some files in a GitHub repository, which you can clone with the git clone command as usual. If you take that and run it, it'll do the same thing as you'd expect it to. Next, we're going to look at some of the files that we downloaded. You can see in the context menu that I downloaded a bunch of stuff. But what we're really just interested in are this HIV reference FASTA and this reads.fasta. And now these files are just example files, so they aren't very big and will easily run within the Colab environment. However, if they were, let's say, a couple gigabytes, then you probably wouldn't want to open it up within Google Colab through the context menu. Instead, you'd want to take a peek at what it has using the head command. Just like in Python, where you can use a head function to look at the beginning of a data frame, and in R, where you can use the R version of the head function to look at the beginning of matrix data, you can use this Unix head command to look at the beginning of a file in the Unix file system, as opposed to cat, where if the file was really large, then cat would print all of it and it would probably crash your notebook. So as you can tell, this reads.fasta contains 3 sequences from some humans. Next, we have the complete genome of some HIV virus. And you can tell that, yeah, this is what it looks like. And even though it doesn't show the complete thing, you can use this command on the HIV reference FASTA, and that'll confirm that it's just one complete sequence, so the entire genome. In comparison, if you use it on the human reads, you can confirm that there's just 3, which makes sense because otherwise head would print out a bit more. Yeah, so moving on. So in order to perform sequence alignment, we'll need tools that are not provided with R, so we can't get them with either through Bioconductor or GitHub or CRAN. So instead, we'll have to download them from various sources across the internet. Thankfully, there are things called package managers, which simplify the installation process and also help manage dependencies. So for example, let's say a certain program wants to run Python 3, whereas another program wants to run Python 2. Normally, you'd have a hard time having both of them running properly on the same system, on the same computer. However, with package managers, you can containerize them in different environments and thereby bypassing the need for like having to deal with conflicts of dependencies. So yeah, so the package manager that we'll be using today is called Anaconda. Has lots of great programs contained with it. And at its core, it has something called a Conda command. Similar to Git, it also has lots of subcommands that are very useful. And this Conda command will do the heavy lifting in terms of package management and environment management. Additionally, because we have limited memory within Google Colab, we do want to be careful of how much we use. So instead of installing Anaconda in full, we'll be installing a streamlined version called Miniconda. That way we save memory and can run larger data sets within Colab without having to pay Google some money to increase the memory limits. So to start, we can download Miniconda from this link. Here we also append dev null to the end of these lines. As you've seen before, here's the wget command, which you can use to get content off the web, so webget. And here's a link to a Unix script, a shell script that someone else has written. So essentially the same as the shell scripts we've written previously in the course, but this is a shell script that someone else has written. And what this shell script does is that it'll install and download the Miniconda. Anyways, and then at the end, there's dev null syntax will send any output or errors from these 3 lines of code and essentially get rid of them. Where since you've been introduced to the redirection operator before, this right here, it'll send, again, the output and the errors to a file called dev null, and anything that gets put in the dev null file essentially gets sent to the void. It's gone, and that helps keep our notebook clean, because otherwise it would create a lot of text. And as before with R, we don't want to have too much text fill up our notebooks, especially when we're downloading things. So in R, remember, we used quiet equals true as an argument for the bioconductor and other download commands. Anyways, just like before with Unix shell scripts, we also use the chmod to change permissions. You can, as before, you can tell that we have permissions to run this as an executable file, because we ran chmod right there. And then, like before, we also use this dot syntax to refer to the current directory, and then to execute this shell script within it. It has some other commands after it, but you don't have to worry about it. They're arguments and options specific to the shell script. But anyways, with that code run, that means Miniconda is now installed. So next, we want to make sure that Miniconda is the latest version. The reason for this is because if you go to the Miniconda website, it'll have like a bunch of different versions and previous versions of Miniconda, and you could get a link from here, but it's faster just to update the the mini Conda through within the notebook. This is also more reliable in general too. The reason you want to do this is so that Conda is able to get the latest versions of the packages, and yeah, helps things run smoothly. So by running this, we have the Conda command here, and then we use the subcommand update, where we're updating the base environment. So you can just think of the base environment as the notebook itself. And we're updating the Conda package. So we're using Conda to update itself. And additionally, we add this yes command here, or not command, this option at the end here, because otherwise it'll expect some user input. And as we've saw in previous labs, sometimes when running commands, they'll ask for some kind of user input, and either you have to input numbers or certain codes or whatever. And to avoid that, we add yes here, because to prevent any issues with interactive terminals not working super well within Google Colab. Okay, so then moving on, we'll now create a Conda environment. So the notebook itself has its own base environment, as I've mentioned, but we want to create a sub-environment within that. And the reason for that is because that way we can install the packages that we want into this specific environment without messing up the notebook as a whole. Additionally, we can have multiple environments where different environments might have different programs that would otherwise be incompatible together. But by having them in different environments, we're able to run them from the notebook. For now, though, we're going to use the Conda create subcommand, and we're going to name this new environment Lab 10 environment. And to avoid the interactive stuff, we just say yes, that yes, we do want to indeed create this environment, and that Conda does not have to ask us twice. And so doing that, you'll get this text, and it'll say to activate this environment, you'll use Conda activate and then the name of the environment that you created. This is normally how you'd activate an environment on, let's say, your university cluster, or your own personal computer, if you want to run programs on that, as opposed to within this notebook where, again, we're using Google's computers. But instead, in order to use this in Colab, we instead want to use source activate, and then the name of our environment. In fact, we want to add this as a header for every code block that we want to use Conda within, or rather this environment. The reason for this is because otherwise the environment will not persist between code blocks. For example, I can show later on that after we install a package into this environment, if we try to access it from the base notebook, it will not be available unless we include this bit of code, source activate, and then the name of the environment. Okay. So before we install stuff, any questions so far?
I have a question.
Sure
If
thing.
we install Anaconda instead of mini Conda, what will change our output of this test? Thanks.
Oh, so the text for creating an environment will essentially be the same. It won't change anything because as this image shows here, it'll essentially be the same because you're using this core Conda command. The only difference would be that you'd have less memory to work with within Google Colab.
But this will influence the results of analysis or not?
No, this will not.
Thank you.
Yep. Any other questions? Okay, sounds like we're good to install. Okay, so as introduced a bit in the lecture, today we'll be installing an alignment program called Bwa, which uses the Burrows-Wheeler transform algorithm to perform alignment. So, like before, we'll want to have this header, so the bash header, as well as the header to access the new environment that we've created. And so we want to install the package Bwa. To get the code for that, you do something like you Google like Conda Bwa, and the first result right here, you can see Bwa. And then it provides the code that you'll use right here. And you can copy and paste that. However, like before, for many of the Conda commands, you want to add yes to confirm that, yes, you do want to install this and that Conda does not have to wait for us. Once you run that code block, it'll start installing and it'll describe the changes that have happened. So here it says new packages that were installed and shows that it downloaded and extracted stuff and just setting things up. A lot simpler than having to install these things yourself. So in order to use Bwa, so that was installing Bwa, and we can call just the command Bwa, and it will provide a help message. So similar to when we called the name git before, it provides a list of the subcommands. In this case, we have Bwa Index, Bwa Mem, and a bunch of other things. You'll see that actually there's multiple alignment algorithms within Bwa. For example, there's Bwa align. However, we won't use that because Bwa Mem, according to the developers, is the latest and best alignment algorithm within Bwa. And by best, that means that out of the reads that you give it, it will have the most success mapping reads to the reference. For example, if you use this align instead, it may be faster or not, but it's been shown that it has less success in mapping reads to a reference. And in that case, then you're using less of your data, and that can influence your analyses. Like, for example, p-values and statistical significance, because if you're using less of your data, then the conclusions you make will have less confidence. Anyways, now let's use this subcommand called Bwa Index. And what this does is it converts the our reference, so the HIV genome in this case, and it converts it to a special data structure that helps with computation. So you can think of it as like, when you're cooking something, sometimes you have to prepare the ingredients before you actually start mixing the ingredients together. That's basically what Bwa does. It prepares the genome by adjusting it a bit. Anyways, we can learn a bit more about the specific options of Bwa Index by calling the subcommand. here we'll want to use this P option, and that will ensure that we can create the special data structure objects within the context folder. Otherwise, it'll make them within the repository, which we only want to access the repository for its files. We don't want to update the repository. So it's better just to have it in the context folder. In addition, this in.fasta here, as the name suggests, is the input reference FASTA sequence that we'll use to index. So
can you repeat once again? Bwa index does what?
So Bwa index will convert the reference genome. So as we saw before, it's a FASTA sequence, and it'll convert it into a special data structure. It's fairly complicated. Like it performs a Burrows-Wheeler transform to produce a special like FE index and yeah. When we run the command, you'll see a bunch of files. And yeah.
And what does it mean?
The index stuff.
Yeah.
Okay.
Option
let me
minus.
send you a link then, since you're interested in that real quick. Actually, where's the? Let me get this one. So this is a good video about it, but this is just if you're interested in it. It's not necessary for this course, and this is more on the side of like if you're trying to create, write your own bioinformatics tools, as opposed to being on the user side of things, where you're more interested in using other tools to help your own domain-specific research. But yeah, so it's essentially lots of math. But yeah. Let's see. So then, now that we called the name of the subcommand and know how to use the subcommand, we can fill in the arguments. And by running this, we'll see that it worked very, very quickly, which is great because our input sequence was very small because we're using a viral genome as opposed to like the human genome or the mouse genome. And here we have different files. Here's the BWA, the Burrows-Wheeler transform. Here's a suffix array, and some other files related to the output of Bwa Index. And so, but once you see those, you know that it worked. Next, we'll use this Bwa subcommand. And so you can call the name of the subcommand to learn about what you can do with it and its various command line options. Here we have IDX base, which represents the prefix of that we specified here. So HIV ref. That's because all these files have that prefix, and this will tell BWA-MEM that here's the special index files, and that it can use those for alignment. Then we have our inputs, FASTQ sequences. Only one is required, but for example, if you have paired-end sequences, it also accepts more here. Anyways, okay. We'll run this. as follows, where here we have the Bwa mem command. We're giving it the prefix. We're giving it our FASTA sequences from the human cells, however that was obtained. And then we're using this redirection object or operator to save it as a SAM file called alignments.sam. If you don't use this, then I'll just print. See, it just prints it out, but it doesn't actually save it to a file. Anyways, now that we have a SAM file, we can use Samtools. And so on that note, we have to install Samtools, much like we installed previously. So with you'd Google something like Conda, Samtools, and then get the link that way. So yeah, and there's the code for downloading it, and here's yes. I guess another thing you could do if you want a specific version of Samtools is that you could type Samtools equals like, and then whatever version number there is. On that note, you can actually check the versions, previous versions that are available by clicking the Files tab. And then you can look at all the previous versions or whatever's listed here at least. And you can see the numbers right here, like 1.8, 1.0. And like it's like the newer version appears to be like 1.9 or something. Or no, 1.14. That's the latest version. And but yeah, if for whatever reason you need to access older versions, then that's how you do that. Like in some cases, I've come across programs where certain functionality was removed in later versions, and you might want to use those functions in the earlier version, even though the developers decided not to update that anymore.
So Cynthia,
Yeah.
about Bwa, it's so this is the function that transforms that that special file into a SAM file. Like it makes
Yes, it does the alignment.
It does the alignment. Okay. And you mentioned that we do Bwa mem and then the prefix. What does that prefix do? Hivref.
This prefix specifies the prefix of the special index files. So again, here we have the Burrs-Wheeler transform and transform and the suffix array. So all of these files are related to the index that we created to help with things computational wise.
So we created with index function that files, and we specified in the Bwa index the prefix hiv /ref, and because we specified there this prefix, then all the files were created with that prefix.
Actually, not in this case, because see, we renamed the SAM file to alignments.sam. We could also have named it to HIV, like human or something. But it wouldn't make sense to call it HIV ref because at this point, the SAM file is an alignment of the human sequences to the HIV reference. So
no, I get that, but where did we get this hivref? Where we created it in the previous function of index, right?
So yes, we specified it in this previous function. However, originally, we got this prefix from the original GitHub repository. So if you check here, then you see it already has the HIV_ref prefix. And we just carried that on through our pipeline so far. Is that clear?
I guess so. Yeah, thanks.
Yeah. For example,
Because I don't understand the meaning of a prefix. Why do we indicate it? I understand why we indicate the file which we operate with, but what does the prefix do? Why we indicate it?
so the reason why we indicated this prefix file here during the index step was that this way it creates the file within the context folder, because this is what we want to use as our project folder. However, for example, if you run this code without the prefix, then it'll put all the files within the Git repository. And as I mentioned before, we're not interested in making adjustments to the repository here. And that way, it makes it kind of messy to have the files of our analysis mixed with the files that are in this repository. So yeah, in addition, you could also use the prefix just to change the name into something you'd like. So like lab 10 files, then if you run this, now we have a new set of index files that have the same data, but now they have a different name, where they're all lab 10 files dot whatever.
And if we don't indicate any prefix, then it will be made into the origin of that FASTA file.
Yes, correct.
Okay, got it. Thanks.
Yep. Okay. So here we used Bwa mem and got our SAM file. And now we want to install Samtools. Like before, it'll do its installation, and it'll tell us the different packages that it changed. And then from there, we can call the name Samtools and get a little help information. And here we can see the subcommands associated with the Samtools command. So our next step is that we'll want to convert the SAM file into BAM format. For example, when running downstream analyses, often programs will prefer the files to be in BAM format, simply because it takes up less memory. And because, as you recall from lecture, BAM you can think of as just the binary version of the SAM file. So in binary, everything's encoded and compressed, whereas SAM is just plain text. In addition, once we've created this BAM file, we might want to look at its information, which would be the SAM format, but in this case, you could actually delete the SAM file afterwards and you'd be fine. It would save memory, or rather storage space. And however, we can't just use the Unix commands that we're familiar with before, because remember, it's a binary file, it's encoded in a certain manner, and it's not human readable. So instead, we'll use the Samtools view command, and then what that'll do is it'll output the SAM format contained within the BAM file. So here we have these different columns, and like here you have the cigar string also mentioned in the lecture, where let's say this first one had it aligned to the HIV reference at position 105, and starting from there, it had 18 nucleotide matches, 1 nucleotide insert, and then 52 matches afterwards. So that's a pretty good match alignment to the reference. So, and then here you have the sequence. Do notice, however, that the header is missing, because SAM files have both headers and the main body. So if we look at just the SAM file itself that we had before, you can see that here's its header, and it tells you different information, like we used BWA for this alignment. It also says where this is from, so an HIV complete genome, which was our reference. And so if you want to preserve that information, then you'll add this -H option to Samtools view. And now we can output the SAM format again, but with the headers included. Anyways, so this data set itself isn't that large. It's just 3 reads and 1 reference, and they each mapped to different places. The 150th position, the 1,700th or so position, and then the 5,900th or so position. And they have no overlap. And so in terms of depth, or in other words, coverage, it's just 1, because we only have 1 set of reads for each of those aligned locations. However, sometimes there'll be situations where multiple reads will align. So let's say, for example, you have a BAM file of sorts, and let's say it has all of these reads, and you want to know at a specific position in the reference how many reads cover that particular nucleotide. So what's its depth? And one way we can get information about all the locations that we're aligned to is with this Samtools depth subcommand. And here you can see essentially what you'd expect, where starting at the 105th position, we have a depth of 1 for the length of the read, this first read, and then starting at the second read, depth of 1. There's a 0 here because as you can see in the cigar string, it had 1 deletion. And so that's the deletion, the deleted nucleotide. But going on, everything else aligned. And then in the final one, you have just same depth of 1 for the third read. Again, though, these numbers would be much different if the input data was more complex. In any case, though, with that, the tutorial version of this assignment is now complete. Any questions? Okay, sounds like we're good for the assignment. So there's not a lot of instructions to the assignment because in this case, things are a bit more open-ended. Whereas in the previous 2 modules, we're trying to introduce you to code that's a bit on the complex side. First by asking you questions about our code, and then asking you to fill in blanks within our code. Now, instead of those big chunks of code, thankfully, as you saw, you can actually run these commands with just a single line of code. So now we're at this point where we can run our own code. And in this case, we're simulating a situation where you want to use a certain tool online and for your own research. And assuming that tool is downloadable by Conda, you'd use what you learned in this lab in order to download and install that program. In this case, we want to use an alignment program, and then you'd be able to run it like we did before with Bwa Index and Bwa Mem. So Yeah. So you'll look for some kind of alignment software that's not BWA, because we already used BWA. But we'll look for something else. And you can also look at the documentation. You can also do what we did before, where we called the name of the command and its subcommands, and it would give us helpful information about how to run them. And then once you are able to align the 3 reads to the HIV genome, like we did with BWA, then you'll use the same Samtools commands to create the BAM file. And the BAM file should be the same as before, because these aren't reads that are hard to align. So yeah, there in the lecture, we did show a couple a couple aligning programs that were mentioned in terms of differential expression analysis pipelines. See, where's like, for example, here's some aligners. So yeah, this assignment is a bit on the open-ended side. You can use one of these, or you except don't use Salmon or Callisto, because those are aren't aligners in the traditional sense. Again, they use a quasi-mapping method, but any of the traditional kind of aligners you can use, and you can look up what aligners exist out there, which ones are available on Conda, and then do what we did before, but with the program of your choice. Any questions now?
I have one. BWA alignment software is used only for transcriptome, or can be used also for alignment of genome?
It can be used for either. In this slide, it's only shown for the transcriptome, because for differential expression analysis, it's the simplest way to align to the transcriptome, because if you're aligning to the genome, recall that during alternative splicing, like in the original genome, you have both the exons and the introns. However, if you want to align specifically for differential expression analysis, or rather using Rna-Seq data in general, then you'd want to use what are called splice-aware aligners, which understand that there can be gaps within the alignment. For example, so your Rna-Seq read, for example, in that case, would just be like a straight line. Actually, let's make that red. So it would just be a straight line, because it's just one read. However, in the genome, okay, it needs to be red. In the genome, you'd have, just please see red. In the genome, you'd have maybe this, and then this, and however, this would come from this part, and this piece of the genome would correspond to this part of your Rna-Seq read. So this here is RNA, and this is DNA. And then also in the genome, you'd have intron. So that's what it means by splice-aware aligners. It means that it understands that the entire sequence of your Rna-Seq data doesn't have to be aligned exactly to the genome, that there can be gaps in it due to introns. Does that make sense?
Yes, thank you very much.
Yeah, happy to help. But yeah, yeah, that's what this is referring to in that diagram. But otherwise, you can use BWA and other related aligning software on either genomes or transcriptomes. To start, we'll be using Miniconda again, just like the last lab. However, we'll also be using something called the Mamba, which, like Miniconda, it's a package manager. However, whereas Miniconda and Anaconda, the larger version, is written in Python, Mamba is a re-implementation of that in the programming language C++, which speeds up the package downloads and environment creation by quite a bit, like taking potentially 1/3 the time you'd expect Anaconda to do the task. Additionally, another advantage of using Mamba is that it works a bit smarter and can bypass certain errors that would cause Anaconda to fail at certain steps. So we'll install it using Conda because that's all we have at the moment. And once it's installed, actually, copy this. Run that code. And then so we'll install it with that code, and then we'll create an environment called Lab 11. Here we use Mamba instead of Conda as our command. There, you can basically think of them as being equivalent for most intents and purposes. The only output difference being that Mamba is a lot faster. So with that, we will begin transcript quantification with Semin. So as mentioned in previous lectures, Semin uses a quasi-mapping technique in order to obtain count data for differential expression analysis purposes. Yeah, unlike BWA last time, which we used to align reads base by base, this time we'll be using this quasi-mapping method.
It is still a method of alignment, right?
Kind of. It doesn't do alignment in the traditional sense, where you, as the name suggests, align reads to a reference. As was described in the lecture a bit, the goal with quasi-mapping isn't to align the reads, as in know exactly where in the reference certain reads will map to. It's instead to figure out which transcripts are associated with which reads. So you don't need to know the exact location in a transcriptome of where read aligns. You just need to know that it's generally associated with a certain transcript. And that's okay, because ultimately what we care about are the transcript counts in terms of expression.
So it like only determines if that read is coming from that gene from which that transcript was made.
Correct.
Mm-hmm.
Okay. Any other questions? Okay, moving on then. Well, as before, we need to activate the environment as a header for every code block. And here we'll install Semin, specifically version 0.12.0, because some of the other versions, with Conda at least, can have various errors that prevent it from getting installed and being run within Colab. Whereas this version works. Sometimes you have to fiddle with it. Like, for example, if you go to the Semin page for Anaconda, you can see all the versions, which we looked a bit at last time. However, one thing you can try is that, let's say the version that that Conda or Mamba, which Mamba is better, whichever it downloads, if it doesn't work for a certain reason, then you can look at some of the older versions or just any other different version and test out which ones work and don't work. And you might find some success in that way. At least that's for how it worked for Salmon. Anyways, upon installation, it'll give you details about what it's installing and the progress with it. So yeah. Then once that is finished running, we will verify whether Salmon was properly installed. So backing up a bit, we'll create the environment and we'll install Mamba. Or we'll install Salmon through Mamba. Anyways, then by running this code, we get some help information for Salmon. We can see that there's multiple subcommands: index, quant, aliven, swim, and quant merge. All we'll use for this lab today are the index commands, subcommand, and the quant subcommand. Similarly to BWA, we need to index our reference. And before we're able to perform, in this case, pseudo alignment. Anyways, once Salmon is set up, we now need to download a reference transcriptome. So as a reminder, the transcriptome is like the genome, but for RNA. So all the mRNA that gets expressed. And that works because we want to use Rna-Seq data to align to that, and in turn, perform differential expression analysis. Anyways, today we'll be using Arabidopsis thaliana as our reference transcriptome. And the reason for this is that its genome itself is quite small. And within Google Colab, we only have so much space to work with too, at least without shelling some money to Google. And so that's why we've chosen this. We could have just as easily chosen other organisms with small genomes like yeast, with Saccharomyces, or, well, technically anything. Even a virus genome would work if you can get enough RNA-seq reads for it to do the to run differential expression analysis. But anyways, so we'll access this reference transcriptome from this URL, and we'll download it with this curl command. It's a kind of equivalent to wget in that it does also download, and curl in this case stands for client URL. Additionally, if you notice the beginning of this URL here says FTP instead of HTTP. FTP is short for file transfer protocol. So it's used specifically for file transferring. We'll see a bit more about that later. So we'll download this file. And next, we need to prepare the reference transcriptome using Salmon index. Here we have the Salmon command, index subcommand. We use this -t option to specify our transcriptome. here you'll see it also has the extension .gz. So what that does is, or rather what that means is that the reference transcriptome is compressed to be smaller. As such, it takes up less space within Google Colab or any other computer you downloaded it to. Additionally, that we don't have to decompress it or anything beforehand because Semin Index will already recognize that it's compressed. Additionally, we use this -i command to name the prefix. So this is the directory that we're creating called erythral index. Or just athal index. So yeah, when it runs, it'll give this output. And
what does the -t do? Excuse me.
the -t.
Yeah, dash.
So that argument, this option will, this flag represents that you need to put the transcriptome file as a command line argument right after it. Yep.
And transcriptome file, this is a FASTA, yes, FASTA file,
yes.
But I understand it is compressed and with this extension GZ, right?
Yes.
Okay. And can we use, if we use just FASTA but not compressed, we need to change somehow, I think, some flags, you call them.
Or
we
just
need
these options.
So we need to look at the options depending which input file we have as a reference transcriptome, FASTA or compressed. We can change these options in the command.
You won't need to change this option specifically because it'll recognize either a FASTA file or a compressed FASTA file, and both will work.
Both will work.
Yes.
Okay.
Just the compressed version is preferred because, as you can see already, we're running a bit low-ish in terms of disk space on Colab. But anyways, I clicked on it and now it's trying to load something. Well, okay. That just means I have to restart everything. Okay. So yeah, we created the index, and the index itself looks like this. I have all these different files, similar to how when we ran BWA last module, it created the Burroughs-Wheeler transform file and the suffix array file. But here we have a bunch of different files. But yeah, so moving on, we've used Semin to index our transcriptome reference, and now we need RNA-seq reads to pseudo-align to that reference. So we'll use Mamba to install this program called SRA tools. This package is used to download SRA files from NCBI's sequence read archive. In addition, if you use the program called Fast QDump or Faster QDump, those are two different programs, but they do the same thing, then it will convert those into FastQ files during the download process. So we'll download that, and on the side, I should run that. So we'll set that up, and we can verify that Faster QDump is inside the toolkit that was just installed by running Faster QDump help. Compared to Fast QDump, you basically always want to use Faster QDump because it's both faster and it has better design principles. For example, you have to mess around with certain command line options with Fast QDump, but here, if you want to download something, you just type in Faster QDump and run a session from NCBI.
And Faster QDump, this is a function of
from SRA tools?
From SRA tools.
Yes.
Yeah. Okay.
Okay.
It is used for pseudo aligning. This faster QDump function.
It's used for downloading.
For downloading.
Yes.
Okay.
Like for example, if you go to the NCBI SRA, and just look up some kind of sequence, that's looking kind of weird. Okay, there we go. What it essentially will do is equivalent of clicking send to and then file and it's equivalent to downloading the FASTA file for a specific run. So yeah. There's ways to download it manually. However, within Google Colab, it's best to use the command line, especially if you want to download, let's say, tens or hundreds of files. You wouldn't want to do that manually. So instead, you use Faster QDump, which again is just the best practice for downloading any files from SRA.
Yeah, thank you.
Yep. Any other questions?
It only works for NCBI, right? Because we only indicate the SRR number and it only works for NCBI runs.
Correct.
Okay.
Okay. So the reads that will align to Arabidopsis selenia will come from a study that investigates the effect of cyclic AMP on triggering a stress response within the plant. If you're curious, you can read a bit more about what this is, but the point is, there's lots of these studies out there, and the journals will require them to post their data, RNA-Seq data somewhere. And here in this case, their data made it onto SRA. So like in the previous lab, we'll have 3 control samples and 3 treatment samples. And that's good in this case because we already don't have that much space within Google Colab. So in order to download, you'll use the faster qdump command, followed by the run a session as an argument. Note that it says SRR. There's other things on NCBI like SRX and other SR something. They all represent different things, but what we want is the SRR specifically. So yeah, these will actually take like in total maybe like 30 minutes to download, maybe 40 minutes. I don't remember how long it took exactly, but it makes sense.
It's very long. I started when we start the course, and just the second one is downloaded.
I see. But yeah, takes a long time since you're downloading millions and millions and millions of reads. And each of these themselves are 400 megabytes, which is on the smaller side. If you look on SRA, a lot of the sequences, or rather a lot of the read libraries there are like 1 or 2 gigabytes large. But anyways, once those files are downloaded, we'll quantify expression with semnquant. So this is the other subcommand that was part of semn that we'll use today. So like in module 2, where we introduced using Unix scripts, we'll use a for loop here to run semnquant on every FASTA file that we've downloaded. So all 6 of these. Again, it'll iterate through every FASTQ file where this asterisk wildcard represents the ID before it, but it can be anything because that's what a wildcard is. Then each file you can call with the name file. And here we'll get the file name, just the entire file name. And then we'll extract just the ID with this bit of code here. So whereas the file name would say like SRR or SRR something something dot fastq, the ID would just be the SRR part. Then we'll echo a little message to say which sample is getting processed. And then after that, we'll run semnquant with these arguments where I, in this case, represents the directory that has the index. So a thal index. And then you need this -L option to describe the library type of the Rna-Seq data. If you don't know that, that's okay. You can just put a capital A here and semnquant will automatically detect the library type. In addition, we have -R for our reads, which are contained in the FASTA file. And then we also have this option called validate mappings, which, as the name suggests, it validates the pseudo alignment as a little extra step to improve the quality of the pseudo alignment. In addition, we don't want to get too many messages, so we use quiet, and then we use -O to format the output. So we'll store it in a folder called quants, and the name of the subfolder within quants will be the ID name. anyways, that'll also just go over all the files and take a fair bit. Definitely not as long as the download process, but it will take a fair bit. And once it finishes, you'll have the quant.sf files, which, as we saw previously, we can use for differential expression analysis. So the code here is the same as you've seen before, pretty much. We enable R within Google Colab. We download packages that are necessary, including TXI meta to convert the quant SF files into something DSeq2 can use. Then, in addition, we'll have a metadata file, except last time the metadata file was in the Git repository that we cloned to get our input files. However, we're not getting our files from a Git repository, we're getting it from NCBI. So that means we have to write our own metadata. And here, we use this format where we have the variable sample name, and what we want to put in the sample names variable is a vector, which is what they call it in R. You can think of it as a list in Python terms, but it's from the data structure side, it's fundamentally different. And the syntax for that is C, which is short for collection, and then parentheses. So you have a collection of all these strings, which have to be in order because they will refer to the samples. So the first sample should be in the first slot, the second sample should be in the second slot, and likewise. In addition, we'll create another variable called sample type, and using the arrow redirection operator, we'll store this vector, which says what kind of category the sample is, whether it's part of the control or whether it's part of the treatment. Then we'll create an R data frame, which was just kind of glossed over last time, but here we'll talk about in more detail, where by calling this data frame function, you can then specify the columns of this data frame, where we have an ID column with the sample names, we have a category column with the sample types, and then just this additional arguments to make sure it works properly. Because we do not want these strings to be factors. So that's why we put false. So yeah, upon calling the name of this data frame, we can see its contents, and it's pretty straightforward.
Once again, what does the factor mean? Strings, factors, false.
So it will interpret the strings in a different way if this were true. It would interpret them as factors. So reviewing the R documentation, you can see here that what the strings as factors argument does in a formal sense is that it asks whether should character vectors be converted to factors. And we don't want that, because then they won't be strings anymore. And what we want are strings because the point here is to label our samples, not to represent them like, for example, as a numeric data.
Okay, good, thanks.
Yep. Like, for example, if instead of control and treatment, let's say you wanted to compare different kinds of treatments, and let's say you had treatment 1 and treatment 2, if you had strings as factors as true, then these numbers might get converted into number type data structures. Whereas we don't want that because we want them to stay strings. So yeah, that's why this argument is here.
Got it.
Anyways, continuing on, much like before, we want to get the absolute path of our quant.sf files. And then we'll create yet another data frame containing the absolute path, the ID, and then its category. Okay. Next, we'll do the same step as before, create a summarized experiment type object, and then we'll import that into and convert it into a DSeq dataset type object with this command. And then once we have the DSeq dataset, we can run the core DSeq commands, and that'll process that, from which we can extract a results table. Then with this results table, we can plot a volcano plot, which for this data set looks like this. And then we can apply the transformation to the data for a PCA plot, as well as a heat map. So same stuff as before. On that note, that is the end of the tutorial section for this lab. Any questions?
But we didn't have any alignments.
No alignments.
No alignments.
Yes, because we used Simon, which doesn't do alignments.
Yes.
Yep.
Okay.
Any other questions? Okay. Moving on to the assignment now then. So essentially the purpose of this assignment is to let you run this pipeline, but on your own data. And by your own data, it'll be publicly available transcriptomes and RNA-seq reads from the same organism. Likewise, if you ever need to do differential expression analysis in the future, then at that point, you'd already know how to do that using Salmon, TXI import, and DSeq2. So yeah. So to start, as some hints for where to get the transcriptome and the RNA-seq data, one place you can get transcriptome data is from this link, the sequence and annotation downloads from the UCSC genome browser. And then here at the top, there's a bunch of different organisms. And you can pick something you like here's a bunch of worms. Maybe they have small genomes, because again, when you run this yourself, you do not want to go over in terms of disk memory, because then your analyses will fail and the notebook will crash. But anyways, once you've selected whatever organism you want to use, you can then click on annotations and then click on the file server link. That'll take you to this special looking website. It's very minimalistic on purpose, because what it essentially represents is a file system similar to the file system that you can view on the left side panel of Google Colab. However, in here, what you'll want to look for are the transcriptome files. So let's see. Let me go to yeast. Because I checked out yeast earlier, so there it is, Estervecier. So then here, you look through these directories for the kind of like FASTQ transcriptome file. And it turns out that, at least for this organism, it's in the NCBI Rna-Seq, which itself is a database. And then here we can see the .rna.fast extension. So there is our transcriptome. You can click on it, it'll download it to your computer, but that's not what you want. Instead, you'll right-click it and copy the link address. And then you can use either wget or curl to download it into the notebook. So for example, for this link that I just got, let's see. We can call this file the same name. So, and this should work. Yep. Here it downloaded the FAST file. And we can take a peek at it. With the head command. And here is some gene that's present in yeast. You can Google what specific gene it is. And here we see that it's a gene for a transcription factor within the yeast. But yeah, so yeah, all these organisms here should have files like that. You could even check out COVID, see if there's anything. See if we can get a reference file from this. Nope, not that. Well, in any case, it might be there, it might be not. If you can't find a file like that, you can just try a different organism. But yeah, just because I showed the way to do it for yeast, you don't have to do it for yeast. And any organism that isn't the organism that was shown in this tutorial. So the Arabidopsis thalassius. But yeah, that is one way to get the file. And so then once you've chosen your organism and have the transcriptome from it, you'll then want to get some RNA-seq reads. And one way to do that is to use this search string on NCBI SRA. So what you'll do is you'll change the genus and species to what you're looking for. So just can pick some organism. Copy that. And once you've replaced the name of the genus and the species, you can then search for sequences on the SRA database. Notice at the end that we have control, and that's help find samples that have the word control in them. And the reason why we want the word control in our search is because we want to find sequences from studies that have a clear treatment and a clear control. So for example, here's something from an says untreated control. And you can get the SRR, or in this case, ERR ID, and use faster QDump to download that. However, we wouldn't pick this one because this one's 3 gigabytes and that'll take forever to download into Colab. And you might run out of space too. So that would not be optimal. But if you can find something that's looking a lot smaller, I don't know why I put that asterisk there before. So here's something that's kind of okay, like 400 to 500 megabytes. That's what's currently used in the tutorial. However, it doesn't look like this one clearly expresses which one the control is. Oh, wait, no, it says in this study, the treatment was this, and then the control was that. So to look at all the samples associated with the study, you can then click all runs under actually study. This one right here. And then look through it and okay. So here we go. Here's 3 files containing the treatment of sorts, or rather the yeast that expresses this PS-AVH gene, I assume, and then the wild type version of the yeast. And the file sizes are fairly reasonable, all around 500 megabytes, which still will take a while. So if you can find something like 200 megabytes, that will speed up the download. But so then what you do at this for something like this is you'd get the run sessions, and like before, you'd create an R data structure to label them. So like you'd have ERR here, ERR there, but for each of the samples, and then you'd label them either control or treatment, depending on what they are. So yeah, that is what you'll be doing today. So looking for your own transcriptome and RNA-Seq files for an organism, and then running a fairly complete differential expression analysis pipeline. And I say fairly complete because all the important, super important data processing steps are there. However, when you're downloading sequences just off the internet like this, you'd want to make sure to include a quality control step. However, there's multiple ways to do that, and it would like require its own kind of lecture, which today was the last lecture. So, but yeah, we won't be talking about the details of quality control, but if that might be something you might want to look into further in the future, if you want to use publicly available data sets. But just for running the tools themselves for differential expression analysis, that is something that we've shown the tutorial here, and you'll run it yourself. Any questions?
Can you show again the, you know, when you said that you get to that fairly simplistic looking page? I'm trying to get you that on any organism, but I don't know why it's not. I don't get such a page. On
Okay.
That one.
So which organism did you select?
I tried for SARS, but also for C. brigazid, you know.
What I see. That one. C. brigazid. So let's see. We have the annotation file. You can go to the file server. And do they have anything? Again, it just might not be here because even though it'll have annotation files, it might not be an annotation file in FASTA format. So yeah, it looks like you wouldn't be able to use this organism.
Okay. But when we find it, if it's any FASTA file, we know already that it's an annotation FASTA file?
Yes. Before, well, yeah, pretty much, because all of this is annotation data.
So the samples we only get from studies. There is no samples here.
Yeah, there are no samples here. These are all reference sequences and other kinds of information.
Got it, thanks.
And reads files should be only FASTA files. Reads files.
The read files?
Yes,
yes.
read files. Only FASTA, because
yes.
Then we have this one FASTQ files.
Oh, yeah, no, yeah, that'll work.
Okay,
like,
we need to search for FASTA files.
Yes, in terms of the annotation for your reference transcriptome. The reason why it's FASTA and not FASTQ is because these are reference sequences that, like, they've been assembled and constructed and curated by other individuals. And so the purpose of those files isn't to provide you with quality information related to a sequence. It's just to provide the reference. That's why they're in a simpler format, FASTA format, instead of FASTQ.
But this site doesn't have any search input or something like this.
Say that again?
Search for a genome, I can't do it, right? I have to click one by one and try to find a match.
Yeah. You're also welcome to look for other websites to find. Like, there's also a website called iGenomes that provides transcriptome information. However, it's a bit complicated somewhat because you have to open, like, for whatever reason, it doesn't open in Chrome. But if you have Microsoft, their Internet Explorer, then maybe you can get to the page. I don't know, the files are technically here, but I've had trouble myself trying to download them from this specific site.
Could you send the link?
Yep. How about you, Aditya? Do you have any experience downloading from iGenomes?
No.
Yeah. So actually, we can try this incognito window in Google Chrome. I think it may work.
Okay. Oh, still the same error. But yeah. So yeah, because that website's a bit buggy, that's why I suggest using the UCSC genome database. But yeah. Any other questions?
The question, there are other programs that we can use when we will work with the larger files, because Google Colab didn't have so much space.
So you'd run that either on your university's computing cluster or your own computer through terminal. So as mentioned a bit before, if you have a Macintosh computer, like Apple computer, then you have an application called Terminal in which you can run these kinds of commands. For Windows, however, you'll have to go to the Microsoft Store and install Ubuntu.
Thank you. And another question is the output of this laboratory. We need to upload our notebook and a BAM file. I know that the BAM file is a result of alignment. In Rna-Seq, we didn't.
Yeah. Ignore the BAM file thing. Yeah. So yeah, ignore the BAM file. We just want a copy of your collab notebook. That's carryover from last assignment. Oops.
Thank you.
Thanks for catching that.
I'm trying to finding the genome for Escherichia coli, but without any success.
I say
I found the genome for zebrafish, I think, but when I search for some studies, how do I know for which species? Like we only say zebrafish, that's it? Or?
It'll have the scientific name of zebrafish. I think it's like Danio something. Yeah, Danio rario there.
Oh, I see.
Additionally, keep in mind that we're getting the transcriptomes, not the genomes, because we want to have transcriptomes to align RNA-seqs against.
And how do I understand that one will be a transcriptome?
Because it'll be under the annotation section.
Oh yeah, it was under the annotation.
Yep. If you click this genome section instead, then you could get genome files. Like here, there's GTF files for genes, but oh, this also works. So here you have if you see an ref mRNA.fasta.gz file, that's also Ref-seq mRNA, which is essentially the transcriptome.
I see.
But yeah, any other questions? By the way, Aditya, it seems like you might have had something you wanted to say. You can feel free to take the stage now. If
Yeah, actually, yeah. So basically, I want to say that like if you want to know all the what do you say, all the arguments for each of this library, then you can go to the documentation of that. For example, if you go type salmon on Google Chrome, then it will go to the documentation and it will show all the arguments that it has. And if you want to know the function of like how is the tool working, then you can also go to the through their source code and like and all these things as well. All of these are open source software.
and I'm gonna stop sharing my screen right now. But again, let me know if any of you have questions.
Yeah, I actually got some queries from one of the student. I don't remember his name. Yeah, he wanted to discuss some of his questions of the previous lab, as far as I know.
Did you get this question just now or like through an email?
Actually, through an email a few days before, I guess. Yeah.
Gotcha.
Yeah, if that person is available, then we can create a another window and then talk about that.
Or it was me who reached out to you, I think. Yeah, and I will, as Cynthia, you told us, I guess yesterday about the hours that you're going to have, that we as students can reach out and then discuss what kind of problems we have, so then we can submit our labs.
Yep. Yeah, we still have to decide on the office hour times, but yeah, once we have figured everything out amongst us TAs, we'll send out an announcement to the group email.
Okay, thanks.
Yep.
I found a study with control groups and treated, but it has only one ERR run, and I can't find to be multiple.
Well, if that's the case, then that study is probably not good. However, to double check things, would you like to share your screen?
Yeah, sure. I still don't have permission.
Oops. Okay, the settings are changed.
Mhm. This is the study I found, and this is where it says embryos treated with organofluoride and against control groups.
Yep.
And this is the run.
Okay, that's just a single run.
Yeah.
Scroll back up.
Okay. 1 second.
Okay. Go to where it says study and then
Yeah.
all runs.
Oh.
Because that page is just for a single run. Okay, here, yeah. Multiple files. Control, low exposure, high exposure, mid exposure.
Yeah.
Though, just a warning, those files are a bit larger than normal. So you can expect to wait a bit longer.
Okay, thanks.
Yep.
Cynthia, can I use the resources? Oh, okay.
Yes.
Okay. Yeah.
Yeah. Any two groups that you can expect to have different expression.
Yeah, it seems to work, but now I have to find the genome. Transcriptome.
Yep. Oh, I see. It's Drosophila, the fruit flies. I'd have to look in that specifically, but for Drosophila, you can get the, where is it? Yeah, I already found the Rna-Seq file for the transcriptome in the, for Drosophila, which you can check through this link.
Thank you.
In terms of helping you all find the transcriptome files, I added a bit more information to the Lab 11 public version. So you can check the public notebook for a bit more information. Since if you made a copy of it already, then it won't be changed. But the important thing is that you can click either of those links on the UCSC page, and the files that you're looking for are either labeled like mRNA FASTA.gz or something like sequence reference like seq and TBI ref-seq, and anything that refers to mRNA, and it is a transcriptome file, most likely. There will also be some information in the README part of the files on this website, so you can confirm that it is indeed a transcriptome from there. For example, to screen share a bit, these files would be transcriptome files.
Okay, got it.
Yep.
When we run that for loop to create the quants, right?
Yes.
Why does it create a separate folder where it puts it? Is it automatic, or did you specify anything to do it in the quants folder?
Because I specified a quants folder and then a subdirectory within that, the salmon quant subcommand automatically created that directory. So scrolling down to that part. Screen sharing. So this part right here, where there's the O option, with the argument quants I, and then slash, then the ID. This is what told Salmon Quant to create this quants folder.
And it named it quants by default.
I named it quants because I wrote quants
okay.
here.
You said no. Yeah, I see.
Yes.
Okay. Thank you.
Yep.